doctors detect diseases like HIV or COVID using a small sample? The answer is a powerful technique called ELISA. In this video, I will explain in simple way so that you can understand it clearly and also it will be useful in exams. So, hello all, welcome to the channel Sarantraj classes. Today we are going to learn about ELISA enzyme linked immunosorbent assay, one of the most powerful analytical technique in pharmacy. So in this video, I will cover how it works, different types, principles and procedures, and its applications. Make sure you watch till the end, you will get complete clarity. So moving on to the topic, what is ELISA? ELISA is a immunoassay used to detect and measure the specific substances called antigen or antibodies. Consider consider doctors are having a urine sample or biological fluid sample. In that particular sample, there are several antigens. So in order to detect how much of antigen present in that sample, this method plays a vital role in that place. So in simple term, it answers two questions. One is the sample present, is the substance present in the sample and how much it is present. Moving to the principle of ELISA, the principle is based on the antigen antibody interaction. When they bind together, an enzyme reaction produces a color. So the more color means the more concentrations. Moving on to the requirements of ELISA. To perform ELISA, we need some of the components, some such as biological components and then chemical components and then instrumentation. First, the biological components. The sample. The sample may be a serum sample or saliva or urine cerebrospinal fluid. And we need antibodies. In this case, we have different types of techniques of ELISA. So in some technique we use only the primary antibody. In some cases we use the secondary antibody also. So generally antibodies are the primary and secondary antibodies. And then enzyme label. HRP or alkaline phosphatase linked to that antibody. So the chemical components. So these chemical components such as blocking agent, bovine serine, albumin or casein, and then washing buffer, PBS, T, and substrates such as TMB, OPD, and H2O2 system producing color for measurements. So this by using the biological component and chemical components, we can conduct the reactions but the how much of the color produced is basically determined by the instrument called microplate reader. So these reactions are conducted in a micro titter plate, 96 well polystyrene plate and micro pipettes. So sequence of ELISA. So in that, first immobilization. So we have the vessel such as the microtiter plate. In that vessel we are coating that either antigen or antibody. So and then we are adding if we coating antigen on that microtiter plate, then we are adding the specific antibody. This specific antibody binds that of the antigens. So once it forms a complex, we are adding the enzyme linked. So these particular enzymes bind to the head of the antibody. So the whole entire process is, we can consider at the bottom we have antigen. Above it antibody is linked to that antigen. With that antigen enzyme is linked. So these complex forms. So remaining we have antigen, antibody and then enzymes that are spreaded into the microtetraplate. So in order to remove that, we are using the procedure called washings. After wash out the procedure, we are adding the substrate reactions such as the signal generations. This substrate reacts with the enzymes. These enzymes makes the substrate to turn colorless compound in the colored compound, and the color is measured using the micrometer. So stages of ELISA. First is antigen coating. As I said previously, antigen binds to the plate and then sample addition. Patient sample with primary antibody is added and then enzyme linked antibody. Enzyme linked secondary antibody is applied. So substrate reaction. Substrate is added and color changes is noted colorimetrically. So types of ELISA. So there are four different types of ELISA. One is the direct ELISA and then is the indirect ELISA and sandwich ELISA and competitive ELISA. First, moving on to the direct ELISA. In this method, antigen is coated on the plate and detected using enzyme linked primary antibody. So first we have having the, for example, consider it bread. So that bread as a antigen. On that we are adding the antibody such as jam. So first the antigen is below, and above that we are adding the jam. So fruit jam, that fruit jam act as a antibody. So in above that we are adding another bread that acts as a enzyme. This is replicates the direct ELISA. So this method is simple, fast method, but lower sensitivity due to the signal amplification. So moving on to the next type, indirect ELISA. So first here antigen is attached to the plate, then primary antibody binds. So here we are adding first the antigen, above that we are adding the antibody, in that which initially forms the antigen antibody complex. With that, we are adding the secondary antibody which is linked to the enzymes. So this secondary antibody binds to the primary antibody. The primary antibody binds to the antigen. So it makes the more complex. So this method is more sensitive and flexible, commonly used to detect the antibodies in serum. Moving on to the third topic, third type, sandwich ELISA. Capture antibody is coated on the plate. So before direct and indirect ELISA, we are coating the antigen. Here we are coating the antibody to the plate, and we are binding the antigen to the antibody, and then second enzyme is linked to that of the antibody detects. So highly specific and sensitive, ideal for measuring antigen concentration in the complex samples. So, and moving on to the final type, competitive ELISA. Sample antigen compete with the labeled antigen for binding to an antibody. So this technique is signal intensity is inversely proportional to the antigen concentration in the sample. So this is a pictorial represent for the direct ELISA, indirect ELISA, sandwich ELISA, and competitive ELISA. So this is the overall content of the techniques we are used before. So what is coated in direct and indirect ELISA? We are coating with antigens, but in sandwich ELISA we are coating with capture antibody. But in competitive ELISA we are adding either antigen or antibody. So what is detected in each techniques? So in direct ELISA, direct ELISA and sandwich ELISA and competitive ELISA, we are detecting the antigen concentration present in the sample. But in indirect ELISA, we are detecting the antibody concentration present in the sample. With that antibody concentration, we are indirectly detect the how much of antigen present in that sample. So sensitivity is competitive ELISA is moderate sensitivity, but indirect ELISA is highly sensitivity. Regardless specificity, indirect ELISA is highly specific, and sandwich ELISA also, and competitive ELISA. Compared to these four types, very highly specific is the sandwich ELISA. So suitable for small molecule, none of first three methods is not suitable for the small molecule, but competitive ELISA is highly suitable for the small molecules. So background noise is high for the indirect ELISA, and the time required is much longer for the sandwich ELISA. So cost of the experimental techniques much higher for the sandwich ELISA and lower for the direct ELISA. So typical application for the each type is direct ELISA has the rapid antigen detections. In indirect ELISA we are detecting the antibody, but in sandwich ELISA we are detecting the cytokines and various biomarkers, and competitive ELISA we can analyze the drug assays and the hormones. So data interpretations, we have measuring the concentration with respect to the absorbance. So the enzyme reacts with the substrate which produces the color. The intensity of the color is subjected to the UV spectroscopy, spectrophotometer, that gives the absorbance, which is with that we can know the concentration by plotting with the absorbance concentrations. So nature of the results, in this method we can get result in qualitative and quantitative method. So in qualitative, what kind of either the presence or absence of antigen or antibody we can detect, but in quantitative we can measure how much of antigen, antibody or hormone present in the sample. So, and then moving on to the advantages of ELISA, it is highly specific and highly sensitivity, quantitative and qualitative analysis, rapid and time efficient method, cost efficient technique, non-reactive and safe producer. In radioimmunology, it uses the radio-labeled isotopes, but here we are using the simple uh, enzyme linkage. So here there is no radioactive emissions. So and it is also advantages due to the high throughput screening capability and widely applicable in clinical and research fields. And its disadvantages of the ELISA is cross reactivity and non-specific binding. Either it may bind to the well of the microtiter plate, limited sensitivity at very low concentration. We cannot detect the how much of concentration of analyte or the antigen present in the sample. Enzyme instability over a period of time it may degrade, so that we can specific sensitivity of the technique may be lost. And then matrix interference from the biological sample and time consuming and multi-step procedure, quantification and reproducibility variability. So applications of ELISA, HIV detection and hepatitis B detections, COVID-19 in hormonal assay and anti autoimmune disorder markers. In pharmaceutical application, we ELISA is used in the therapeutic drug monitoring and vaccine antibody titration, cytokine estimation, bioanalytical method validation, and stability studies. So conclusions, ELISA represent a far powerful integration of immunology and analytical chemistry, offering precise scalability and clinical relevance. Its ability to deliver accurate quantitative data has made it indispensable in diagnostics, research, and pharmaceutical quality control. As biomedical science advances, ELISA continues to evolve, reinforce its position as a foundational analytical